Hello everyone, welcome to the lecture. In this lecture we'll talk about the metabolic pathways for carbohydrates. So we discussed carbohydrates till now, we discussed enzymes and vitamins, we discussed proteins. Now let's enter into metabolic chemistry. So let's start with the lecture outline. So in this lecture we'll talk about metabolism and energy, the important coenzymes in the metabolic pathway, digestion of carbohydrates, process of glycolysis, the pathways for pyruvate, and 6 glucogen synthesis and degradation, and gluconeogenesis, where we synthesize glucose. So let's start with, before we enter the lecture, so be sure to revise the following topics: identifying the D and L Fisher projections, describing the enzyme action, classifying enzymes, identifying the factors that affect enzyme activity, and the role of cofactors in the metabolic processes. So let's start with metabolism and energy. So metabolism involves two types of reactions. One is catabolic reactions, the other is anabolic reactions. Catabolic reactions are basically, the term catabolic itself basically says that you're basically cutting down large complex molecules to provide energy and smaller molecules. Anabolic reactions are used they use ATP energy and build larger molecules. The combination of both catabolic and anabolic reactions combined are called as the metabolic reactions. Now let's start with the stages of catabolism. So before we start catabolism, remember the two processes. Catabolism is where we're trying to break up the molecules that are large and produce energy and make it smaller. So there are three stages that catabolism works in. So the stage 1 is the digestion and hydrolysis breakdown of large molecules to smaller ones that enter the bloodstream. So think of these as sugars, any general molecules that are taken from the larger starch molecules. For example, let's say if you eat a bunch of bread, you're basically taking down starch, and the starch is broken down into sugar molecules. The sugar molecules are then going to enter the bloodstream. Stage 2 is within the cells. These degradation breaks down the molecules to 2 or 3 carbon compounds. So you're basically taking the little larger compounds and further breaking them down. And stage 3 is the oxidation of the small molecules in the citric acid cycle and electron transport to provide the ATP energy. Remember that catabolic processes are exothermic processes, which means that these are the processes that release energy. So these are the three stages. The first stage is digestion and hydrolysis. For example, if you take proteins, they undergo the process where they break down and become amino acids. Polysaccharides become glucose, fructose and galactose. Lipids become fatty acids. So remember that these are all broken down before the cell membrane, and only the smaller molecules will enter the cell membrane. Once it enters into the cell, it undergoes stage 2, where it undergoes degradation and some oxidation to smaller molecules. Amino acids undergo transamination to form pyruvate, acetyl-CoA, and some of them enter the citric acid cycle. If you take sugars, they generally undergo glycolysis and form ATP. And the further downward products create pyruvate, and pyruvate then enters into the citric acid cycle. And fatty acids undergo beta oxidation to finally enter the citric acid cycle. And citric acid cycle is where the third process takes place, where they undergo oxidation to form carbon dioxide and water and energy for ATP synthesis. So notice that in the entire process there are 3 ATP molecules being produced. So you have one ATP molecule being produced under glycolysis, one ATP molecule being produced in the citric acid cycle, and one ATP molecule produced by the electron transport and oxidative phosphorylation. So these are the 3 ATP molecules that are being produced in the cycle here. So let's start with the cell structure of the metabolism. So cells in plants and animals are generally called eukaryotic cells. So these are cells that contain a nucleus that contains the DNA. So in animals, you have a cell membrane that separates the materials inside the cell from the aqueous environment surrounding the cell. Now the cell nucleus will contain the genes that control the DNA replication and protein synthesis, which we discussed in the earlier lecture in nucleic acids. And you have cytosol, the fluid part of the cell that contains the electrolytes and the enzymes that catalyze chemical reactions in the process. So most of the reactions generally takes place in the cytosol, except for the process of DNA replication, which takes inside the nucleus. So metabolic reactions occur at different parts, different specific sites within the cell. So for example, electron transport mechanisms and ATP production are generally done in the mitochondria. Protein synthesis is done in the ribosome. DNA replication is done inside the nucleus. Cell membranes are where the initial products that are broken down into smaller molecules enter. Cytosol is where you find most of the enzymes. Lysosomes are the molecules that take in the products that are created and deliver it to outside the cell. So let's start with each of the structures. So first start with cell membrane. So cell membrane basically separates the content of a cell from the external environment and contains the structure that communicates with the other cells. Next you have cytosol, is the fluid part of the cytoplasm that contains the enzymes for many of the cell's chemical reactions. Next you have a lysosome. This contains hydrolytic enzymes that digest and recycle old cell structures and create old cell structures. So mitochondrion, which contains the structure for the synthesis of ATP from energy producing reactions, or most commonly called as electron transport chain reactions. You have the nucleus, which contains the genetic information for replication of DNA and synthesis of proteins. You have the ribosome, which is the site for protein synthesis using the mRNA templates. So let's start with the molecules that are involved in metabolic processes. First molecule is ATP, the short form for adenosine triphosphate. The ATP molecule contains of a base, adenine. So notice that the base here is adenine. You have a ribose sugar. Notice this is a DNA, so it has a ribose sugar. So this is an RNA product, RNA molecule. So it contains a ribose sugar. And you have 3 phosphate groups. So these are the 3 phosphate groups. These hydrolyze. So if one of the phosphate groups is removed from ATP, it creates ADP. And if you further break it down, it creates AMP, which is adenosine monophosphate. So triphosphate becomes diphosphate, diphosphate becomes monophosphate. And these bonds are what release the energy of an ATP molecule. So ATP molecule holds energies in the form of high energy phosphate bonds. So the hydrolysis of ATP to ADP releases about 7.3 kilocalories per mole. That's about 31 kilojoules per mole per each single molecule. So if an ATP molecule undergoes hydrolysis, it creates ADP and a phosphate, and it releases energy. Now the hydrolysis of ADP to AMP also releases the same amount of energy, 7.3 kilocalories per mole. Now ATP is the main molecule that drives the reactions. For example, if you take catabolic processes, which are the energy producing processes where you take food, the food that you take is stored in the form of ATP. Then you have anabolic reactions, which are energy requiring processes, for example like running, sprinting. These they get utilized here, and under the utilization it releases the energy. So it undergoes hydrolysis and releases the energy for you to do that work. So one process is producing the energy and the other process is requiring the energy, but notice that there is a net reaction. The net reaction is always going to release energy. This here is the reason why metabolic processes are net energy producing reactions, are not net energy consumption reactions. So try to use the same principle now. Pause the video right here and try to answer these questions. Next, let's talk about the important coenzymes in the metabolic pathways. So one of the first enzymes is coenzyme A or CoA. It's made up of several components. So first of them is the pantothenic acid, which is vitamin B5. You have phosphorylated ADP, and you have amino ethane ethiol. So you have amino ethane thiol, you have pantothenic acid, and you have the phosphorylated ADP molecule. This entire molecule is called as the coenzyme A. So let's start with metabolic reactions. So metabolic reactions that extract energy from food involve 2 types, both oxidation and reduction reactions. So here oxidation involves the loss of hydrogen or electrons by a substance or an increase in the number of bonds to the oxygen. Reduction is the gain of hydrogen ions and electrons or the decrease in the number of bonds to oxygen. So in both of these types of reactions, we use coenzymes. These coenzymes are required to carry the hydrogen ions and electrons from or to the reacting substrate, because the hydrogen molecules themselves, the hydrogen ions themselves, do not have an easy way to transfer from one place to another place. So think of coenzymes are assisting processes. These are the ones that that deliver the hydrogen ions from one place to another place. So a coenzyme here that gains hydrogen ions and electrons is reduced, whereas a coenzyme that loses hydrogen ions and electrons is oxidized. So remember that here, if you gain hydrogen atoms, so the molecule is being reduced, the coenzyme is being reduced. If you are losing hydrogen atoms, if you are losing hydrogen ions, let me write it this way. So if you are gaining hydrogen ions, reduced. If you lose hydrogen ions, you are being oxidized. So oxidation involves either loss of electrons, loss of hydrogen, gain of oxygen, or producing energy. Reduction involves gain of electrons, gain of hydrogen, loss of oxygen, and requires energy input. So remember that oxidation reactions are the ones that produce energy. Reduction reactions are the ones that require energy input. So let's start with the first coenzyme. So coenzyme NAD+. NAD+ is the common coenzyme that you find in many pathways in your body. So the short form for it is the NAD+, but the actual name is nicotinamide adenine dinucleotide. So it's an important enzyme which is the vitamin niacin, in which the vitamin niacin provides the nicotinamide group, which is bonded to a ribose, and the nucleotide adenosine triphosphate is created. So it is required for the dehydrogenation reactions, where that produces carbon-oxygen double bonds in such reactions such as where we convert alcohols to aldehydes and ketones. For example, if you take ethanol, so ethanol, to remove the hydrogen on the oxygen, we need to use NAD+. NAD+ here gains the hydrogen and becomes NADH. So resulting in the formation of CH3, C double bond of H. So it converts ethanol into ethanal. So an aldehyde, so it basically converts the alcohol into aldehyde. So the name process itself, the enzyme used here is alcohol dehydrogenase. So remember that dehydrogenation basically means that we are removing hydrogen, and here you're doing that for an alcohol. So the enzyme is the alcohol dehydrogenase. Next, the coenzyme NAD+. The oxidized form of NAD+ undergoes a reaction when you have a carbon atom in the nicotinamide ring, reacts with two hydrogen atoms, leaving one of the hydrogen atoms. So this is the actual form of the molecule when it is oxidized, NAD+, where this bond is one that accepts the hydrogens. This is the reduced form where it has accepted two hydrogens. So this is the idea behind NAD+ molecule. Next you have NADP+. NADP+ is a nicotinamide adenine dinucleotide phosphate. So the only addition here is that to the sugar you are adding an extra phosphate. So that phosphate is what is going to be, this is commonly used in anabolic reactions, most commonly in lipid and nucleic acid synthesis. So it's similar to NAD+, except that the 2' OH group is replaced with a phosphate. So NADP is the oxidized form, NADPH is the reduced form. So remember the difference between NAD+ and NADP+. So the only change here is the 2' OH group. So in place of the 2' OH group, we have a phosphate group. Next is the coenzyme FAD. FAD is flavin adenine dinucleotide. So it contains two molecules, ADP and riboflavin, riboflavin which is vitamin B2. So it undergoes reduction when two nitrogen atoms in the flavin part of the FAD+ coenzyme react with two hydrogen atoms, reducing it to FADH2. So remember that NAD only takes in one hydrogen, but FAD actually takes in two hydrogens. So FAD in its oxidized form is not charged. So it can accept two hydrogens on two of the nitrogens. So it can accept it here and here. But so once it accepts, you will see the two hydrogen bonds with the nitrogen bonded here. So what is the function of this coenzyme FAD? So it generally participates in reactions that produce a carbon-carbon double bond. So most commonly ends up reducing to FADH2 with the aid of the enzyme succinate dehydrogenase. So succinate is this structure where you have a diacid structure. Undergoing FAD+, it creates fumarate where it creates a double bond. So it removes this two hydrogens will bond with FAD and create FADH, and it creates a double bond. Next, let's talk about coenzyme A. So coenzyme A is derived from a phosphorylated ADP and pantothenic acid, which is vitamin B5, bonded to an amide bond to amino ethane thiol, which contains the SH reactive part of the molecule. So this is the one that reacts with the entire molecule. So we write the short form here as HS-CoA. So HS-CoA, HS here is the one that ends up bonding with other molecules. So if you take an acetyl group and bond it with CoA, we create acetyl-CoA, which is a thioester, which is one of the most common compounds that you see in the citric acid cycle. So what are the important functions of CoA? So it prepares for small acyl groups such as acetyls for reactions with enzymes, and it's also involved in the production of energy-rich thioester, acetyl-CoA, which is used in the citric acid cycle in the production of ATP. So what are the types of metabolic reactions? So the metabolic reactions which take place at body temperature and physiological pH always require enzymes and often coenzymes. So remember that enzymes are the ones that change the rate of reaction, and coenzymes are the ones that assist the reaction and move the molecules around easily for the reaction to occur. So for example, oxidation reactions, you have dehydrogenases where you have NAD+, NADP+, and FAD. Reduction reaction also undergoes a dehydrogenase where you end up creating NADH, NADPH, and FADH2. You have hydration reactions where the enzyme is hydrase, there is no coenzymes involved. If you take the transfer of a phosphate group, you have phosphatase and kinase. The coenzymes are ATP, GDP, and ADP. You have transfer of an acetyl group, which use the enzyme acetyl-CoA transferase. The coenzyme is CoA, and you have hydrolysis, which involves protease, lipase, and synthetase. The coenzyme there is ADP and GDP. So pause the video right here and try to answer these questions. Now let's get into the main topic, the digestion of carbohydrates. So carbohydrates are the ones that begin in the mouth. Then you have proteins, begins digestion in the mouth, and then proteins in the stomach and the small intestine, and the lipids in the small intestine. So this is how the digestion occurs. So let's start with stage 1, is the digestion of carbohydrates. So carbohydrate digestion first begins in the mouth, where you have your saliva, which produces the enzyme salivary amylase, which breaks down the polysaccharides into smaller polysaccharides called dextrins, maltose, and some glucose. Now then in the stomach, the partially digested starches enter the acidic environment, where the low pH stops for the carbohydrate digestion. Next is the stage of digestion in the small intestine. So what happens inside the small intestine? Inside the small intestine, the pH is a little higher, is about 8, where you have enzymes produced in the pancreas hydrolyze the remaining dextrins to create maltose and glucose. So these enzymes produced in the mucosal cells that line the small intestine hydrolyze the maltose, lactose, and sucrose. So then you have the resulting monosaccharides then getting absorbed into the intestinal wall into the bloodstream. So this is how sugar enters your bloodstream. In the liver, you have hexoses, fructose, and galactose are converted to glucose, and the primary energy source of the muscle contractions in your body, red blood cells, and the brain. Remember that hexoses and hexoses such as fructose and galactose cannot be directly absorbed by your body, so they have to be converted into glucose for them to be used in your body. So what happens in the mouth? First you have polysaccharides undergoing splitting into dextrins and maltose and glucose. In the stomach, there's nothing much changes because of the low pH. Once it enters the small intestine, the dextrins here, under using the pancreatic amylase, create maltose. The maltose, under using the enzyme maltase, splits it into glucose. The lactose molecule using lactase creates galactose and glucose. You have sucrose molecule under the use of sucrase enzyme creates fructose and glucose. The fructose, galactose go into the liver, then get converted into glucose and enter back into the bloodstream. And that is the primary source of energy for your cells. So let's start with the next process, which is glycolysis. Glycolysis is the process of oxidation of glucose, where the glucose that enters the bloodstream enters our cells, where it undergoes the degradation in a pathway called glycolysis. So the glucose obtained from the digestion of polysaccharides is degraded into glycolysis to pyruvate. So the stage 1 you have polysaccharides, stage 2 you have monosaccharides. The monosaccharides then enter into the cells where it undergoes the conversion of glucose into pyruvate. So what is glycolysis? Glycolysis is the reaction, that the metabolic process that takes place in the cytosol of the cell, and it's a metabolic pathway that uses glucose, a digestion product. So in this process it undergoes degradation of the 6-carbon glucose molecule to create 3-carbon pyruvate molecules. So you have the 6-carbon molecule C6H12O6, which undergoes first phosphorylation, where it undergoes the formation of glucose 6-phosphate and or fructose 6-phosphate. Then under further phosphorylation it creates fructose 1,6 biphosphate, and under the final process it creates dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So in the reaction here from 1 to 5 of glycolysis, so you need energy to add the phosphate groups to the glucose. So whenever you see this reaction, it means that this is a reaction where the ATP molecule is being used. So all of these molecules is the ATP molecule. So these are ATP molecules. So there are 2 ATP molecules that are being used in this process, and glucose is then converted into 2 3-carbon molecules. Next, the 2 3-carbon molecules now in the reaction 6 to 10 of glycolysis, you have energy obtaining processes where the NAD creates NADH. The 1,3-biphosphate glycerate then produces 2 ATP molecules, and finally you have the 10th reaction which also produces 2 molecules, finally creating the pyruvate. So you have the first start with glucose, then gets converted to glucose 6-phosphate, and then undergoes another reaction to form fructose 6-phosphate. The fructose 6-phosphate then becomes fructose 1,6 biphosphate, and then creates dihydroxyacetone and glyceraldehyde 3-phosphate. And in the further reaction, the reaction undergoes the formation of 1,3 biphosphoglycerate, and further reaction from 1,3 biphosphoglycerate, it creates the 3-phosphoglycerate where it produces 2 ATP molecules. Then at that undergoes the formation of 2-phosphoglycerate and undergoes dehydration reaction where it removes the water molecules. Then finally creates phosphoenol pyruvate, and finally the phosphoenol pyruvate creates pyruvate where it creates 2 ATP molecules. Notice the amount of ATP molecules that are being produced. There are 2 ATP molecules here, and there are 2 ATP molecules here. Now notice that here we are using 2 molecules, 1 and 1, 2 ATP molecules here, and we are producing a net of 4 ATP molecules. So we are producing 4 ATP molecules and we are using up 2 ATP molecules. So in total you have 4 ATPs that are inside and 2 ATP that are being used. So let's start with the first reaction in glycolysis, which is the phosphorylation reaction. Phosphorylation reaction undergoes the use of the enzyme hexokinase, where the hexokinase enzyme uses an ATP molecule and converts glucose into glucose 6-phosphate, where it attaches a phosphate group in place of the hydrogen on the 6th carbon. So that's why the name glucose 6-phosphate. So you have a phosphate group that is transferred from ATP to glucose, and the glucose 6-phosphate and ADP are produced. So we use the enzyme hexokinase to catalyze the reaction. Now in the reaction 2, it undergoes isomerization, where you have phosphoglucoisomerase, which creates glucose 6-phosphate to form fructose 6-phosphate, and this glucose 6-phosphate, the aldose from the reaction 1, is converted to fructose, which is the ketose. So this isomerization uses the enzyme phosphoglucose isomerase. In the reaction 3, you have phosphofructokinase, which is the enzyme that converts fructose 6-phosphate into fructose 1,6 biphosphate, where it undergoes the hydrolysis of an ATP molecule, where the ATP transfers one of the phosphate groups to the first carbon, so the 1' carbon, and attaches itself to the oxygen, creating fructose 1,6 biphosphate. So after fructose 1,6 biphosphate, it splits into 2 3-carbon phosphate isomers. One of them is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So it uses the enzyme aldolase, which produces the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And in the final reaction 5, it undergoes isomerization, where the dihydroxyacetone phosphate then gets converted into, all of the molecules are converted into glyceraldehyde 3-phosphate. So these here uses all of the 6 carbon atoms from glucose are contained into 2 identical triose phosphates. Now let's come into the reactions where you produce energy. So first reaction that happens here is the reaction 6, where the glyceraldehyde 3-phosphate under the use of the enzyme glyceraldehyde 3-phosphate dehydrogenase undergo dehydrogenation, where it removes the hydrogens and adds 2 phosphate groups. So you have the removal of, so the additions, you have the NAD plus. So the aldehyde group on the glyceraldehyde 3-phosphate is oxidized to a carboxyl group. So this aldehyde group creates a carboxyl group. Now the carboxyl group then attaches itself to a phosphate, finally creating the 1,3 biphosphoglycerate. In the reaction 7, the 1,3 biphosphoglycerate undergoes phospho. So using the enzyme phosphoglycerate kinase removes the phosphate ion. Removal of phosphate undergoes the reaction where two ADP molecules take up the phosphate here and create an ATP molecule. So an ADP takes one of the phosphates to create ATP, which is the energy producing reaction here. You end up creating 2 molecules of high energy compound ATP. In the reaction 8 again the 3-phosphoglycerate converts to 2-phosphoglycerate. So where it undergoes isomerization, where the removal, where you will see the phosphate group moving from this carbon here to this carbon. So yielding 2 molecules of 2-phosphoglycerate. Now in the reaction 9 it undergoes dehydration, where it undergoes removal of oxygen and hydrogen. So there is removal of this hydrogen here. There is also removal of this OH group here, which ends up creating H2O. 2 H2O molecules are released, where it uses the molecule enzyme enolase, finally creating phosphoenol pyruvate. The phosphoenol pyruvate then undergoes further hydrolysis, further phosphorylation with the ADP molecule, where the ADP bonds with the phosphate, the 2 phosphate molecules, to produce 2 ATP molecules, and you end up using the final kinase, which is the pyruvate kinase, finally creating the pyruvate molecule, which stops the final process of the reaction. So in glycolysis, what are we doing here is we are taking 2 ADP molecules and we are converting it into 2 ATP molecules. This is the net reaction. Remember that we used 2 ATP molecules to add phosphate to glucose and fructose 6-phosphate, and we produce 4 ATP molecules in the energy generation by direct transfers of phosphate groups to 4 ADPs. So there is a net gain of 2 ATPs and 2 NADH, creating 2 pyruvate molecules. So you have glucose with 2 NAD plus, creating 2 ATP molecules plus pyruvate and NADH. So this is the overall glycolysis reaction. So this is the overall glycolysis reaction. Next, what happens to fructose and galactose? So I already told you that fructose and galactose are not directly absorbed in your body. So your body uses it. So other monosaccharides such as fructose and galactose can enter glycolysis after they are converted to intermediates. So what happens is that in muscles and kidneys, fructose is phosphorylated to create fructose 6-phosphate, which then enters into the glycolysis in reaction 3. The molecule galactose reacts with ATP to yield galactose 1-phosphate, which is then converted to glucose 6-phosphate, which then enters the glycolysis at reaction 2. And both of these reactions generally happen in the muscles and kidneys for the fructose and in the liver for galactose. So sugars enter glycolysis in different phases. So galactose enters at the reaction 2. Fructose enters either in the reaction 3 or reaction 4, depending on where the fructose is being converted to different molecules. So how do you regulate this? How does your body regulate glycolysis? So it's generally regulated by 3 enzymes. So the 3 enzymes are reaction 1, reaction 3 and reaction 10. Reaction 1, where the hexokinase is inhibited by high levels of glucose 6-phosphate, which prevents the phosphorylation of glucose. In reaction 3, you have phosphofructokinase, which is an allosteric enzyme, which can be inhibited by high levels of ATP and activated by high levels of ADP and AMP. So pyruvate kinase is another allosteric enzyme, which is then inhibited by high levels of ATP or acetyl-CoA. So these are the 3 enzymes that are involved in the regulation of the glycolysis process. So with that, we end the topic on glycolysis.